Zebra mussels begin reproduction when the water temperature is between 10 and 13 degrees Celsius. The immature planktonic form known as veligers remain in the water column for a period of 2 to 3 weeks, at which point they feed and grow large enough that they settle to the bottom and begin their life as the plantigrade zebra mussels that are familiar to most people.
Recreational diving has also contributed to the spread of quagga mussels, so freshwater diving locations are prime sites for monitoring. Zebras and quaggas are often found together.
They basically both behave in a similar fashion. They both filter plankton. They both have byssal threads. However, it seems that the quagga mussel is able to survive in deeper water depths, and it's also able to attach to soft sand-like substrates, whereas the zebra mussel really needs to cling to hard surfaces like rocks and ledges and so forth.
Quaggas prefer slightly cooler temperatures and can tolerate just about any depth as long as there is oxygen present. Both species prefer to attach to hard surfaces, although quaggas can settle on soft sediment. Now that you know where to look for invasive mussels, and you have chosen a site to monitor, we ask that you try to gather data monthly when the water temperature is warmer than 8 degrees Celsius, or 46 degrees Fahrenheit. For most areas of Pennsylvania, the period would be between April and October. Wear polarized sunglasses to reduce glare and improve vision into the water. When monitoring at a lake or a marina, inspect all hard surfaces. Where zebra and quagga mussels are found together, zebras become denser in shallow water, while quagga tend to dominate in deeper waters. However, both species can be detected by carefully inspecting the wadable shoreline. Focus your search on dark, shaded areas. Be sure and check underneath objects like driftwood and in dark crevices behind rocks.
If you can focus.
Gently run your fingers over surfaces, checking for mussels too tiny to see. Newly settled mussels feel like grit on sandpaper. At this point, the zebra mussels have set and are no longer free swimming. Mussels can attach to a wide variety of objects such as pilings and dock floats, buoys, mooring lines or cable, rocks and concrete, logs or driftwood, vegetation, or just about anything that has been in the water for a long time. Fan surfaces covered with silt to expose mussels. Carefully inspect aquatic plants for signs of mussels. Spend as much time as it takes to examine all hard surface objects. Look until you're confident that no mussels occur at the site. If you are finding snails, you are searching hard enough. Be sure to record your results on the filed record sheet that were provided by the monitoring program. When monitoring for zebra mussels, remember that they feed on algae and eat calcium for shell production, so do better in hard water streams with ample nutrient to feed algae. Sample slower moving hard water streams that have enough calcium to support mussel. Small streams that lack sufficient nutrients or have low pH, including acid mine drainage, are poor candidate streams for colonization. River or creek locations that are not near boat access areas and are not close to an infestation are probably not worth monitoring since the risk of invasion is relatively low. Pick monitoring locations near boat ramps, docks, marinas, and under bridges. Another important place to survey is downstream from a known population of mussels, for example, below an infested quarry or reservoir. In streams and rivers, it is best to flip rocks over to investigate the underside. Examine the stream and river bottom. When you return from the field, be sure to transfer your field data from the field record sheet to the Excel spreadsheet. If you think you found a zebra mussel in a new location, please report it immediately. Call from the field if you can. Make arrangements to confirm the specimen's identification. Place 10 to 15 of the largest and smallest specimens in a jar with rubbing alcohol, or place them in a Ziploc bag and store them in the freezer. Early detection and action can help slow the spread of these mussels, so report new sightings as quickly as possible. There are no known methods to completely eradicate invasive mussels from our waterways without killing other forms of aquatic life, so prevention is our best defense. Posting signs on lakes, streams, and rivers is a great way to alert the public and inform water users of new invasion. Once discovered, responsible visitors can prevent zebra mussels spreading to an uninfested lake or river by carefully inspecting all of their recreational equipment, including boats, jet skis, fishing equipment, and scuba equipment. When you pull your boat or jet ski out of the water, inspect and scrape off any mussels you see, and remove all plants and weeds from the prop, motor, and trailer. Drain your bilge, transom, or live well on land before driving to another body of water. Dispose of your leftover bait in the trash, not in the body of water you're leaving. If you are traveling from a body of water infested with invasive mussels to another body of water, rinse your boat, trailer, bait bucket, fishing rod, and live wells with hot water if possible, 40 degrees Celsius or 140 degrees Fahrenheit. Another option is to take your boat to a car wash or let your boat, trailer, and motor dry for at least 5 days before your next boating trip. This will kill the mussels. So remember to inspect, drain, dump, rinse, and dry. It's against state and federal laws to possess and transport live zebra mussels, even those that hitchhike. Monitoring helps prevent the spread of invasive species while preserving and protecting our natives.
By monitoring your local water bodies for the presence of zebra and quagga mussels, you're protecting your ability to enjoy that resource, you're protecting your own property values, and you're protecting the biological heritage that we all share as Pennsylvanians.
So get involved. Be part of the solution. Join the Pennsylvania Zebra and Quagga Mussel Monitoring Network today.